Good morning. Have you ever really stopped to consider the intricate biological warfare that's constantly being waged inside your body? It's a system of such staggering complexity that it protects you from a universe of threats, from a simple cold virus to far more significant invaders. This, of course, is the domain of immunology, the study of our body's defense mechanisms. In this lecture, we are going to dissect the fundamental concepts of this field, exploring the remarkable army of cells and molecules that are dedicated to our daily protection.
So, here is our roadmap for today. We will begin by examining the expansive scope of modern immunology. From there, we'll delve into the body's two principal defense strategies, the immediate, generalized innate immune system and the highly specific, targeted, acquired immune system. We will then analyze the molecular targets that these systems identify, the molecular weaponry they deploy, and, finally, we will meet the diverse cellular players that orchestrate this entire defense. Let's begin.
Historically, when one spoke of immunology, the context was almost exclusively the body's defense against pathogenic microbes, bacteria, viruses, fungi, and so on. And while that remains a central pillar of the discipline, its scope has broadened dramatically. We now understand that the very same immunological principles and mechanisms are fundamentally involved in a vast array of physiological and pathological processes.
This slide really illustrates the dual nature of the immune system. On one side, it is our most vigilant protector. For instance, the concept of immune surveillance posits that our immune cells are in a constant state of patrol, not just for external pathogens, but for internal threats, like nascent cancer cells, which they recognize and eliminate. This same recognition of non-self is precisely why transplanted organs are often rejected. The immune system correctly identifies the new tissue as foreign and mounts an attack. However, this power can be misdirected. In autoimmune diseases, the system loses its ability to distinguish self from non-self and begins to attack the body's own healthy tissues. In allergies, it mounts an aggressive, damaging response to innocuous environmental substances like pollen. And finally, the critical importance of this system is laid bare in states of immunodeficiency, such as in acquired immunodeficiency syndrome or AIDS. Here, the collapse of the immune system renders the body susceptible to opportunistic infections from microbes that a healthy individual would easily fend off.
Okay, let's proceed to our first major topic, innate or nonspecific immunity. You should think of this as the body's pre-configured frontline defense system. It is termed nonspecific because it does not target particular pathogens, but rather acts broadly against any potential threat. It is an innate because we are born with it fully functional. It is our constant, unwavering first line of defense.
The most basic and perhaps most effective component of this system is a set of mechanical barriers. Think of them as the physical fortifications of the body. The skin, with its tough outer layer of keratinized cells, is a formidable barrier to microbial entry. Internally, our respiratory, gastrointestinal, and urogenital tracts are lined with mucous membranes. These membranes secrete a viscous fluid, mucus, which physically entraps microbes. But these are not merely static defenses. The body also employs dynamic flushing mechanisms. The continuous flow of urine through the urinary tract, the flow of saliva in the oral cavity, and the washing action of tears across the eye's surface all serve to mechanically dislodge and remove potential pathogens before they can establish an infection.
In addition to these physical barriers, we possess a formidable chemical arsenal. These are not passive environmental conditions, they are active antimicrobial agents. Our blood contains a host of these factors. Our bodily secretions, such as mucus, tears, and saliva, contain enzymes like lysozyme, which can break down bacterial cell walls. Human milk is another rich source of these protective molecules. Furthermore, the very immune cells that engulf pathogens, the phagocytes, are filled with a potent cocktail of destructive chemicals designed to degrade any microbe they ingest.
So what occurs when one of these barriers is breached? The result is inflammation. It is a fundamental, non-specific response to tissue injury of any kind, be it mechanical, chemical, or infectious. It is characterized by 4 cardinal signs, which have been recognized since antiquity: rubor, redness; calor, heat; tumor, swelling; and dolor, pain. These signs are the classic external manifestations of the innate immune system mobilizing at a site of injury.
So what is the physiological basis for these 4 signs? The process begins with tissue injury. Damaged cells immediately release a variety of chemical signals known as inflammatory mediators. These mediators act on the local blood vessels, causing 2 critical changes. First, vasodilation, an increase in the diameter of the blood vessels. This increased blood flow to the area is the cause of the redness and heat. Second, the permeability of the vessel walls increases, allowing plasma fluid to leak into the surrounding tissue. This fluid accumulation causes the swelling, which can in turn put pressure on nerve endings, causing pain. But the ultimate purpose of these vascular changes is to facilitate the migration of phagocytic white blood cells from the bloodstream into the injured tissue, where they can begin to engulf and destroy any pathogens or cellular debris.
Another systemic response of the innate immune system is fever. A fever is not a malfunction. It is a highly regulated, strategic increase in the body's core temperature. It is induced by substances called pyrogens. These can be exogenous, like components of a bacterial cell wall, or endogenous, meaning they are produced by our own immune cells. These pyrogens circulate to the brain and act on the hypothalamus, which functions as the body's thermostat, instructing it to reset to a higher temperature set point.
Raising the body's temperature is a multifaceted defense strategy. First, many pathogenic bacteria are metabolically optimized for normal human body temperature, and their rate of replication is significantly inhibited at elevated temperatures. Conversely, the higher temperature actually increases the rate of production and activity of our own immune cells, accelerating the defensive response. Perhaps most elegantly, during a fever, the liver sequesters iron from the bloodstream. Iron is an essential nutrient for many microbes, so this action effectively creates an iron-poor environment, starving the pathogens. At the same time, our own metabolism is ramped up to provide the necessary energy for the heightened immune activity.
Now we come to one of the most sophisticated components of innate immunity, the complement system. This is not a single entity, but a collection of over 30 proteins that circulate in the blood in an inactive state, as proenzymes. The system's power lies in its organization as an enzymatic cascade. When an initial complement protein is activated, it cleaves and activates the next protein in the sequence, which in turn activates many molecules of the next, and so on. This creates a powerful amplification effect, where a small initial trigger can result in a massive, localized defensive response.
This cascade can be initiated through several distinct mechanisms, known as the classical, alternative, and lectin pathways. Regardless of the initiation event, the subsequent amplification cascade converges to produce three primary protective outcomes. First, some complement fragments are potent inflammatory mediators, attracting phagocytes to the site of infection. Second, other complement proteins coat the surface of a pathogen, a process called opsonization, which marks the pathogen for enhanced phagocytosis. And third, the final components of the cascade can assemble into a structure called the membrane attack complex, which literally inserts a pore into the membrane of a pathogen, causing it to lyse and die.
While the innate system provides a crucial immediate defense, it is the acquired or specific immune system that provides long-lasting targeted protection. This arm of the immune response is defined by two cardinal features that the innate system lacks: specificity and memory. It can learn to recognize a virtually infinite number of specific molecular targets, and it retains a memory of those targets, allowing for a much faster and more potent response upon subsequent encounters. Acquired immunity can be categorized into four types based on how it is obtained. The primary distinction is between active and passive immunity. In active immunity, the host's own immune system is stimulated to produce a response. Natural active immunity is acquired following infection and recovery. For example, contracting and clearing the measles virus confers lifelong immunity. Artificial active immunity is the basis of vaccination, where an attenuated or inactivated form of a pathogen is introduced to stimulate an immune response and generate memory without causing disease. In passive immunity, by contrast, the host receives preformed immune components. Natural passive immunity occurs, for instance, when a fetus receives antibodies from its mother across the placenta, or when a newborn receives them through breast milk. Artificial passive immunity involves the administration of immune serum containing antibodies from another source, providing immediate but temporary protection.
The entire basis of specific immunity rests upon the system's ability to recognize specific molecules. These molecular targets are known as antigens. An antigen is any substance that can be specifically recognized by the components of the acquired immune system, such as antibodies or T-cell receptors. To be precise, a molecule must possess two properties to be considered an antigen. First, it must be immunogenic, meaning it has the ability to provoke an immune response. Second, it must exhibit specificity, meaning it can react specifically with the antibodies or cells that were generated in response to it. Antigens are typically large macromolecules, such as proteins or polysaccharides, and are usually foreign to the host organism.
It's important to draw a distinction here. A full antigen, or an immunogen, is a large molecule that can elicit an immune response by itself. However, there exist small molecules, called haptens, that are too small to be immunogenic on their own. They can be recognized by the immune system, but they cannot initiate a response. But if a hapten becomes covalently attached to a large carrier molecule, typically a host protein, the hapten-carrier conjugate now functions as a complete immunogen. The immune system mounts a response to the entire complex, and interestingly, some of the resulting antibodies will be specific for the hapten alone.
Now here is a critical concept. The immune system does not recognize an entire antigen molecule as a single entity. Instead, it recognizes discrete small regions on the surface of the antigen. These specific recognition sites are called epitopes or antigenic determinants. A single, large, complex antigen, such as a bacterial surface protein, can possess many different epitopes. This means that multiple different antibodies and immune cells can recognize and bind to the same antigen molecule simultaneously, each at its own specific epitope.
Having discussed the targets, antigens, and their epitopes, let's now turn to one of the primary effector molecules of the acquired immune response, the antibody. Antibodies, also known as immunoglobulins, are proteins produced by B lymphocytes that circulate in the body fluids and bind with high specificity to the antigen that stimulated their production. The basic structural unit of all antibodies is a Y-shaped molecule composed of 4 polypeptide chains. There are 2 identical heavy chains and 2 identical light chains, linked together by disulfide bridges. Each chain has a constant region, which is similar across all antibodies of the same class, and a variable region, which differs greatly from one antibody to the next. The variable regions of one heavy chain and one light chain combine at the tips of the Y to form the antigen binding site. The stem of the Y, composed of the constant regions of the heavy chains, is called the Fc region, and it is responsible for the antibody's biological effector functions.
There are 5 major classes or isotypes of antibodies, each with a distinct structure and function. IgG is a monomer and is the most abundant antibody in blood and tissues. Critically, it is the only class that can cross the placenta. IgA typically exists as a dimer and is the primary antibody found in mucosal secretions, serving as a key defender at our primary entry points. IgM is a large pentameric structure and is the first antibody class to be produced during a primary immune response. IgE is a monomer found at very low concentrations, but it plays a central role in allergic reactions and defense against parasites. Finally, IgD is also a monomer and is found primarily on the surface of B cells, where it functions as an antigen receptor.
The binding of an antibody to its target epitope does not, in itself, destroy the pathogen. Instead, it triggers several downstream effector functions. By binding to antigens, antibodies can cause pathogens to agglutinate or clump together, making them easier for phagocytes to clear. The process of opsonization, which we mentioned earlier, involves antibodies coating a pathogen, which greatly enhances its uptake by phagocytic cells. Antibodies can also physically block the active sites of toxins or prevent viruses from binding to host cell receptors, a process known as neutralization. Finally, the binding of certain antibody classes, particularly IgG and IgM, to an antigen can serve as the initiation signal for the classical pathway of complement activation, unleashing its potent destructive cascade. Thus far, we have focused primarily on the molecular components of the immune system. But these molecules are all produced and orchestrated by a highly complex and dynamic population of cells. Understanding the origins and trafficking of these cells is fundamental to understanding the immune system as a whole. And this brings us to a final, profound biological question. How does the body generate this incredibly diverse army of cells, each with its own specialized function? The answer lies with a single progenitor cell type, the hematopoietic stem cell, which resides in the bone marrow. Under the influence of various signaling molecules called colony-stimulating factors, this single stem cell type gives rise to all of the cellular elements of the blood through a process called hemapoiesis. This includes red blood cells, platelets, and the entire lineage of white blood cells or leukocytes, from the granulocytes and mononuclear phagocytes of the innate system to the B and T lymphocytes that are the masters of the acquired response. These cells then enter circulation, using the bloodstream and lymphatic system as a highway to patrol the entire body, continuously moving through tissues and filtering through secondary lymphoid organs like lymph nodes and the spleen, constantly searching for signs of invasion. It is a truly remarkable system, a coordinated multi-billion cell defense force, all originating from a single common ancestor in the bone marrow.